بسم الله. نبدأ المحاضرة الأولى تكون عن lipoprotein metabolism. هنتكلم إن شاء الله عن definition and different types of lipoproteins, plasma lipoprotein formation and transport in blood, description of structure of cholesterol, its sources, precursors, function and derivatives, and outline the types of dys-lipoproteinemias, their common causes and complications. First, we start with the definition of lipoproteins, and as their name implies, lipoproteins are clusters of lipids and proteins with different ratios between lipid and protein components. This composition is important for the structure so that they can perform their functions. The most important function of these molecules is to transport lipids in lymph and blood. As from their structure, lipids are hydrophobic and need to be bound to hydrophilic molecules to facilitate their transport throughout the body fluids, especially the lymph and blood. Other functions of lipoproteins include their structural role as they enter the structure of membranes of cells and cellular organelles. For the structure and classification of lipoproteins, and as depicted from the figure shown, lipoproteins are formed of a core of triglycerides and cholesterol esters, which are non-polar hydrophobic. So they are present in the core. And a shell that mainly contains phospholipids and some cholesterol, which are polar and confer some hydrophilicity. Embedded in this shell are protein molecules known as apolipoproteins, and they serve as regulators of lipoprotein metabolism. And to summarize, the inner core is formed of triglycerides and cholesterol esters, whereas the outer coat is formed of apoproteins or apolipoproteins, phospholipids, and cholesterol. For the categorization or classification of lipoproteins, lipoproteins are classified in different ways. They are categorized according to their size and density, which are denoted by their lipid and protein content. And as you see in the figure, the more the lipid content, the larger the size, and the lower the density, and vice versa. So the largest in size is a chylomicron molecule, which has the greatest lipid content compared to proteins or apoprotein content, and it has the lowest density. The smallest in size is HDL, or high dense lipoprotein, with the lowest lipid content and highest apoprotein content with the highest density. Regarding the functions of lipoproteins, as mentioned earlier, their structure helps keeping lipids soluble to facilitate their transport in blood. So they serve mainly as a vehicle to transport lipids from and to tissues. We have the protein components of lipoproteins, which is known as apoproteins. They represent the protein part or protein component of lipoproteins, and they are protein molecules weakly bound to the lipid component of lipoprotein and are exchangeable. These proteins serve different functions according to the type present in the lipoprotein. First of these functions is their structure role, as they are essential components of the lipoprotein molecules. Second, they act as activators of some enzymes involved in metabolism of lipoproteins and their mobilization. Third function is that they serve as recognition particles or recognition molecules to facilitate the uptake of these lipoproteins by specific tissues and cells. The plasma lipoproteins. As mentioned earlier, plasma lipoproteins serve as transport molecules for lipids, and we have two types. The triglyceride-rich type for the transport of triacylglycerols and include chylomicrons, responsible for transporting triglycerides of dietary origin. This means after meals, and they are synthesized in the intestine. Second is the very low dense lipoproteins, which transport triglycerides synthesized in the liver or triglycerides of endogenous origin. Second part is cholesterol-rich lipoproteins for the transport of cholesterol and includes the low density and the high density lipoproteins. Low dense lipoproteins transport free cholesterol and this is the most dangerous type of lipoproteins. Second is the high dense lipoproteins, which transport esterified cholesterol, and this is known as the good cholesterol. For the classification of lipoproteins, we have 5 classes of lipoproteins, 4 of which are outlined in the table shown. They all share the characteristic of containing cholesterol, triacylglycerol. cholesterol ester, phospholipids, and apoproteins, but at different proportions. They differ in the site of synthesis, the apoprotein component, and function. We have the chylomicrons that are synthesized in the small intestine, very low dense lipoproteins synthesized in the liver, low dense lipoproteins which are synthesized in the circulation, and deliver cholesterol to tissues. Disturbances in the levels of low dense lipoproteins results in disturbed metabolism and atherosclerosis. The high dense lipoproteins, or HDL, which are synthesized in the liver and serve to deliver cholesterol from tissues to the liver. The fifth class or fifth category is the intermediate dense lipoproteins, which is between very low density and low density lipoproteins. The apoprotein component differs according to the type and function of lipoproteins, and as we see here in this table, for example, chylomicrons, the apoprotein component is apoprotein A, P48, C123, and apoprotein E. And as an example of their functions, apoprotein P48 serves for the assembly of apoproteins and lipids, and apoprotein C serves as an activator of enzymes and metabolism of this type of lipoprotein. The ApoP100 and the very low dense lipoproteins and low dense lipoprotein serves as a recognition particle for the very low density and low dense lipoprotein. They recognize the low dense lipoprotein receptors on cells. The lipid metabolism occurs in three major sites or three major areas. First of which is the intestine, where the chylomicrons are formed. Second is the extrahepatic tissues, where lipoprotein lipase acts on very low density and low dense lipoproteins, and high dense lipoproteins are assembled. The last site is the liver, where very low dense lipoprotein is assembled and delivered to circulation, and intermediate density lipoproteins and low dense lipoproteins are metabolized. For the apoprotein and their distribution and characteristics in lipoproteins, As we can see from the table shown, we have the apoprotein A1 and C2, which are activators of lipoprotein lipase, responsible for metabolism of chylomicrons to reduce their size. The B48, which targets the chylomicrons to the circulation and liver. The B100, which targets very low dense lipoproteins that contain triglycerides and cholesterol to tissues by binding to the low density lipoproteins and serves as a recognition particle for the receptor. And the ApoE or apolipoprotein A, which is responsible for targeting very low dense lipoprotein to tissues. And this slide summarizes the apolipoprotein distribution with the four main lipoproteins and their functions. As mentioned earlier, apolipoprotein A1 activates lysis and cholesterol acyltransferase. The P100, which functions in triglyceride and cholesterol transport and serves as a recognition particle for the low-dense lipoprotein receptor. The B48, responsible for transport of triglycerides. Apolipoprotein C2, which is an activator of lipoprotein lipase. And apoprotein E, which binds to low-dense lipoprotein receptor. Regarding synthesis of chylomicrons and their assembly, chylomicrons are assembled in the small intestine, in the Golgi and the plasma reticulum of enterocytes, where the B48 apoprotein is added. Chylomicrons transport lipids after absorption to peripheral tissues after passing through the lymphatics, and its level is elevated 2 hours after meals. Very low dense lipoproteins are formed after lipoprotein lipase catalyzed removal of triglycerides from chylomicrons. They transport lipids from the liver to tissues. They have the P100 apoprotein, which is responsible for targeting very low density lipoproteins to tissues. This type of lipoprotein, the very low density lipoprotein, is converted to intermediate density lipoprotein, which represents the fifth group of lipoproteins. And finally, metabolized to low-dense lipoprotein, which is uptaken by receptor-mediated endocytosis in tissues, where its accumulation initiates or favors the development of atherosclerosis if deposited in blood vessels. As you see here, the very low dense lipoproteins are converted to intermediate density lipoproteins and finally to low density lipoproteins, which are taken up by receptor-mediated endocytosis. The most dangerous one of these lipoproteins is the low-dense lipoprotein, which originates by breakdown of very low-dense lipoproteins and intermediate density lipoproteins with loss of the apoprotein C2 responsible for their metabolism and apoprotein E responsible for their targeting. The apoprotein B100 remains in these molecules and targets the low-dense lipoprotein molecules to tissues. The last one is the high-dense lipoprotein, which mobilizes cholesterol from tissues and acidifies it by the lecithin cholesterol acetyltransferase activity of the ApoA1 apolipoprotein. This molecule transports the esterified cholesterol to the liver to be eliminated. We have two tables here that summarize the types of lipoproteins, their apoprotein content and percentage, their lipid content and percentage, and their origin and site of synthesis, as well as their functions. To summarize the process of lipid transport as shown in this figure, shows transport and mobilization of lipids starting after absorption from the intestine. Chylomicrons are transported through the lymphatics by circulation to circulation. They are acted upon by lipoprotein lipase and changed into chylomicron remnants, which are transported to the liver and transformed into very low dense lipoproteins. Very low dense lipoproteins are targeted to tissues after losing some of its triglycerides and changed into intermediate dense lipoproteins and low dense lipoproteins. Then the low dense lipoprotein is taken by tissues. Some of the cholesterol of low dense lipoprotein is esterified to cholesterol ester by the L-CAT activity and transported to the liver in the form of high density lipoprotein to be metabolized. The next point is cholesterol. Cholesterol is an alcohol. It's a white waxy compound that is widely distributed all over the tissues with many important functions. of which is a structural function, as it is a structural molecule of cell membranes and cell organelles. Other functions include the bile acid and bile salts synthesis, steroid hormone synthesis, the recommended plasma levels of lipids, as you see here, for the total cholesterol is less than 200 mg per deciliter. Low density lipoprotein cholesterol, denoted as a bad cholesterol, less than 100 mg per deciliter. The high density lipoprotein cholesterol, or the good cholesterol, is about 40 mg per deciliter or higher, and triglycerides less than 150 mg per deciliter. Where the low density lipoprotein is associated with a high incidence of atherosclerosis and cardiovascular disease, and high density lipoproteins serving a protective role in the circulation. The last part is disorders of lipoprotein metabolism and mobilization. First, we have lipoproteinemia, hypolipoproteinemia, or the abetalipoproteinemia, a defect in loading the ApoB100 on the lipid component with accumulation of triglycerides in the liver due to defective triglyceride transport. Second disorder is a fatty liver, where there is increased hepatic sense of triglycerides and reduced mobilization of lipids from the liver. Second category of these lipoprotein metabolism disorder is the hyperlipoproteinemia. We have two important types. Type 1 hyperlipoproteinemia, due to lipoprotein lipase deficiency, which is responsible for the metabolism of lipoproteins. Second type is type 3 hyperlipoproteinemia, which is due to deficiency of the apolipoprotein E that serves as a recognition particle for the low dense lipoprotein receptors. There is also a high incidence of hypercholesterolemia and atherosclerosis with these two types. As you see here in this slide, these represent the laboratory findings in different types of hyperlipidemia as manifested by the turbidity of serum samples 12 hours postprandial. This table summarizes or lists some genetic causes of dyslipidemias that are considered as primary causes with defects in apoprotein synthesis or receptor defects. And finally, we have some causes of hyperlipidemias, which are secondary hyperlipidemias or secondary disorders, the most important of which are diabetes mellitus and hypothyroidism, which are associated with disordered lipoprotein metabolism and increased cholesterol and triglyceride levels in the serum. By this slide, we conclude the lecture for lipoprotein metabolism and their disorders. Thank you.